Good morning, or afternoon, I guess. Or no, still morning. Thank you. I know the comment before, the music was great, but really what I want is I want that announcer on my phone because, boy, that voice is outstanding. Thank you for OIS for inviting, and I just want to say, first off, I'm sorry to hear that Dr. Tauber and Dr. Maxey are not with us today. Wish them both good health and hope they get better soon. I'm here to talk about Tearsolutions. Tearsolutions is a Virginia-based company, and it has basically been developing a dry eye solution. I don't like the term dry eye. I'm going to use ocular surface disease. I'll tell you a story if anyone wants to hear at some point about dry eye. But lacretin is the protein that we are looking at, and in specific, a 19 amino acid fragment, which is the active portion of lacretin. Lacretin was discovered at the University of Virginia, which is where I am now, and it was the result of an unbiased assay looking at over 1,500 proteins and figuring out which protein was deficient in dry eye disease. And I think the subtitle of this slide says it all, which is, if you are deficient, you have the disease. And so at the university, we've been looking at every typical and atypical form of dry eye that we can find, all the way up to analytics. And in fact, they are all deficient in this unique protein. So that's the first bit of information. Obviously, the question is, where do you find lacretin? And lacretin is made primarily by the lacrimal gland and also the meibomian glands. It has not been found in the goblet cells yet, although we're still looking. Corneal physiology clearly identifies that when you look at the lacrimal functional unit, the return to homeostasis is strongly dependent upon lacretin. And we'll show you some data that looks at this. In fact, this very confusing slide is really the pathway for the prosecretory mitogen that lacretin is involved in organizing and in making sure that we have a homeostatic mechanism. There are any number of ways that you can have lacretin malfunction, and that may represent the many different diseases on ocular surface disease. We are looking actively at all of them. Critical to understanding is that it binds Syndecan-1, and that seems to be a critical pathway along with the GPR8 that has been found to also be affected. Now, lacretin and lacrapep are naturally occurring. So I want to say that this is a naturally occurring protein. Lacretin has some activity, but lacrapep, the 19 amino acid cleavage form, is found in normal tears. So this is a normal occurring protein. It does provide homeostasis, but as you can see, what has happened through the work of Dr. Gordon Lowry in cell biology at university is that he has formed a consortium and studied many different parts of the lacrimal functional unit, including and not limited to neuronal growth. And we've seen both neuronal regrowth and repair, which is in process. Actually, it has been published by Nancy McNamara and is going to be published again through some of the consortium. What has happened thus far, so we've taken it from a cell biology perspective and started to put it in clinic. We ran our first in-human trial. It was a unique trial. It was primary Sjogren's patients. And when I say primary Sjogren's, it took us 33 months to find 204 of them. It was a painful process, but it was necessary because we wanted the most severe of the severe to figure some of this out. And in fact, it was hugely helpful. We have identified specific improvements particularly in fluorescein staining and in symptom. And as you heard before, sign and symptom is critical in an approvable drug. We have obtained it in the first in human, sign and symptom at 14 days. And when you look at very particular signs and symptoms, you can identify specific signatures for this drug. The slide, which is a mouse slide, is actually treatment of an airy mouse. It's a knockout model at the University of California. And what it does is basically within 3 weeks, these mice develop severe sicca, really potently severe. On the slide next to it is the same mouse literally 3 weeks later, and this is with human lacretin. This is not with mouse lacretin. So we took human lacretin, treated these mice. We had about 26 mice that this did. They all look like this. And in fact, this slide is responsible for me losing 2 investigators at my investigator meeting for primary Sjogren's because they threw down their gloves and said, okay, I don't want to investigate, I want to invest. And they did. What you can see on the right is the sign and the symptom. It's highly statistically significant improvement. And in fact, as has been shown with many of these trials, the worse the disease, the more profound the improvement, which is very helpful when you're trying to reestablish homeostasis. Again, this is not an anti-inflammatory. This is not a pro-secretory, let's increase the lacrimal gland and see what the Schirmers do. This is curing, returning homeostasis. It's a big statement. I said the word cure. Safe, well-tolerated, really no adverse events to speak of, minimal stinging and irritation as you would expect for a protein. First in human publication finally made it to the clinical journals, and this came out in August. It is still only available electronically. It will be in cornea in written format within the next months, I'm told. And we are looking forward to a rapid motion into a phase 2 dose ranging study to perfect some of the things that we think we need to perfect for commercialization. So in summary, in 22 seconds or less, new chemical entity, novel MOA, clear path to market, and as I said, a restorative, naturally occurring protein that's aimed at restoring the ocular surface disease. Thank you.